Thank you very much, Masood, and for that lovely introduction. And I'm going to introduce my fellow panel members in a moment, but I just think, just to follow on from that, I think one of the things that has been very clear from the session so far today is that we are really at a watershed moment, I think, in terms of dementia research. There is obviously an absolutely growing and urgent need to address how we deliver trials in this area, and as well as support our clinical population. And I think there's a real opportunity in the UK because obviously we have an incredible data set at our disposal through the NHS. And also, as we're coming out of the back of COVID and COVID research and our successes in COVID research, I think we are at a time when people are willing to look at novel ways of managing clinical pathways, gathering clinical data, and also clinical trial design. So with that, I'm going to introduce, as I said, my panel members and ask them to say briefly their background as well by way of introduction. So Martin Landry, if you could start.
Yes, well, thank you and good afternoon. I think we've all met earlier on today, so you know something about me: clinician, clinical trialist, epidemiologist, deputy director of the Big Data Institute, and chief executive of PROTAS, the not-for-profit organisation furthering the development of large-scale clinical trials.
Thank you, Martin. Thor Ostenfeld, you're next on my screen.
Hi, Vanessa. Yes, I'm Thor Ostenfeld. So I'm the lead for clinical development in the neuroscience department at AstraZeneca. And that's part of the early stage R&D organization in AstraZeneca. My background's in NHS, internal medicine, anesthesia, quite a lot of critical care as well. I retained some clinical sessions at Addenbrooke's Hospital, but for the last 20 years, I've been based primarily in the biopharmaceutical industry, GSK, now AstraZeneca, engaged with the translation of preclinical drug discovery relevant to neuroscience indications into early stage clinical research. And one of the key challenges there, of course, is how do you undertake high quality, meaningful, so-called proof of concept trials in neuroscience and in neurodegenerative research, in particular. High quality by design, conduct, size, and at reasonable cost, so that we can give confidence to investors and other stakeholders to invest further in a clinical development program. So
thank you, Thor.
There you have it.
Carrie Rubel, would you like to introduce yourself?
Hi, thanks so much. It's a pleasure to be here as part of this roundtable discussion. I'm Carrie Rubel. I'm a director in fluid biomarkers at Biogen, and I also serve as the biomarker lead supporting the aducanumab program. Since 2019, I've been with Biogen leading efforts developing, deploying novel fluid biomarker assays, largely in support of Biogen's clinical trials in Alzheimer's disease, but also some other neurodegenerative disorders. Before coming to Biogen, I was a research biologist in neuroscience at Merck Research Labs. There I was working in novel therapeutic target ID and also advancing exploratory biomarkers in Alzheimer's. I also have extensive experience in regulated bioanalysis. I worked previously as a program lead doing that type of work at Bioagiletics Laboratories. So thank you very much. Looking forward to the discussion.
Thank you. And lastly, Samantha Benham, Hermets, please.
Hi there, also delighted to be joining this discussion today. My name's Sam Benham Hermets. I'm Director of Policy and Public Affairs at Alzheimer's Research UK. My background is in charities, so previously at Age UK, also at Alzheimer's Society and now at Alzheimer's Research UK. So very much advocating for the breakthroughs in research to benefit people affected by dementia. So as John Gallagher mentioned in the last session, advocating for a dementia medicines task force to look at how we can speed up progress in clinical trials. So yeah, very much looking forward to discussing that today.
Thank you very much. And as Masood said at the start, this roundtable discussion is focusing on new paradigms for clinical trials in dementia, from genes to blood-based biomarkers. So we're going to kick off with some questions that we've prepared beforehand, but please do put questions in the chat for the panel, and we will come to that in due course. So for me, I guess one of the key questions that's come out of the meeting today is how we use biomarkers to direct clinical trials, and particularly what biomarkers should we be focusing on. So in terms of, we should be focusing on blood biomarkers, imaging, CSF, both in the clinical population, if we want to characterize them and make them trial ready, plus also track trajectories in real world populations, but also in the clinical trial population. So I'm going to open this question out to Martin, first of all, if I can. What are your thoughts on that very big question?
Well, I mean, I think there's a couple of things, isn't there? I mean, the first is thinking about biomarkers and risk, and then perhaps thinking about biomarkers and response and outcomes. First, and even before I start that, one thing is to think about how one collects and analyzes biomarkers. It's often tempting to use biomarkers as a sort of a part of your screening and eligibility, to select people who have a biomarker above some threshold, or positive versus negative, or whatever it might be. And there's a few challenges with that. I mean, the first of all is that's means you have to screen a lot of people who are the wrong side of the biomarker divide line, and you throw away. That costs a lot of resource. Second thing is it's time consuming, particularly if the biomarker is on, if it's an imaging biomarker, for example, and you've got limited access to scans, or whether it's a laboratory biomarker and one's thinking about the costs and time taken to assess that before you get the results back, before you can continue with the trial. So I think on both of those levels, think very carefully before limiting. On the, do you really need to throw half the people away? Are you selecting a biomarker because you think that your treatment will only work in those who are biomarker positive? Or are you thinking because it might work somewhat better in those who are biomarker positive? If the latter, in particular, then recruiting a broader group of the patients might actually be extraordinarily helpful, because if you only recruited people who had a stonkingly high cholesterol, you'd never treat the vast majority of people who have an ordinary cholesterol if you're trying to prevent heart disease. So biomarkers can be a very good way to make a common disease rare. There might be some regulatory or financial implications for that, incentives for that, but probably not from a population health basis. The second thing is to say, actually, if you biobank the samples, then there's an awful lot you can work out what biomarkers you really want to do after the event. And once you've got a trial and you can see a clear positive result in your trial, let's hope, then you can start to explore whether the results might, in a post hoc exploratory way, because that's what it is, you can then start to explore to what extent the biomarkers would help you guide towards people who might do better or might do worse. By the time you've done that, number one, you don't measure a whole load of biomarkers when your trial is actually going to be negative overall. And number two, biomarker technology will have moved on. And number three, biomarker technology will have become a lot cheaper. So I think there's a lot of arguments there for not being unduly restrictive about what biomarkers you might measure at baseline. Perhaps I'll stop there.
No, thank you, Martin. And again, I think just pragmatically, from my point of view as a clinician, I think we just, as you said, need to get these processes going and collecting samples and having discussions with clinicians and the public about the utility of collecting those samples. But I'm going to hand over to Tor. I know this came up in one of our sessions as well, about the kind of heterogeneity of potential samples in clinical trials and how biomarkers can be useful in that.
Thanks, Vanessa. Yeah, I mean, we did have a discussion around, particularly in the early stages of clinical research, do you want a more homogeneous or more heterogeneous population? And there are arguments for and against, but I completely buy into Martin's sort of pragmatic approach to this. I mean, certainly for from the drug development, drug discovery perspective, it's tempting to put all your bets on maybe one or two biomarkers that you think are going to be most relevant to the mechanism that your drug is working on. But I wouldn't suggest throwing other biomarkers away, because they serve an important purpose for retrospectively characterizing your population. When you get to the end of the trial, the whole thing may not work, but it could be that there were some interesting findings in particular subgroups that you can characterize better if you've got an array of biomarkers. And the technology, as we heard this morning from Charlotte Toyneson, has moved on to allow us to measure clusters of biomarkers that may reflect where a patient is on the AD continuum, if I can use that phrase. So, I'm still in two minds about the homogeneity or heterogeneity of early stage so-called proof of concept trials in particular. It can work both ways, but it's a difficult game to play when you're instructed to do a small, at reasonable cost study that will give us confidence to invest in the really big trials downstream. That's the challenge that I have in AstraZeneca. But biomarkers are going to be critical to this. And I should say it's not all about disease biomarkers for stratification or for identification of comorbidities. We also need, from the drug development perspective, good biomarkers of target engagement. Because it's the first question I get asked at the end of a trial is, how do you know you engage the target? And you'd be surprised just how many industry clinical trialists have come up with, well, we don't really know. We don't really know whether we got the right dose because we couldn't measure the target engagement properly.
Thank you, Zol. Yes, I think that's an absolutely important question. And as you said, what duration do you need to do a study for in order to pick up that level of target engagement? Getting an alternative industry view, Carrie, would you like to make comments?
Yeah, absolutely. Yeah, I just add to that point, I think, in terms of the target engagement and thinking beyond patient selection. All of these biomarkers, I think, they have different degrees of evidence generation behind them over the years. I think all, particularly thinking about CSF measures of amyloid positivity, amyloid PET, tau PET, tremendous evidence generation has gone on behind these, as well as efforts to standardize. And it's particularly important that we think, as we go through the stages of drug development, what are the particular biomarkers we really need to support at this point in time to answer the questions we need to advance. And I do think, while these imaging and CSF biomarkers are going to probably remain tremendously important for quite some time, the emergence of both blood-based biomarkers are very exciting. And there's a lot of momentum in this field right now, which also is very exciting. But we do need to get to that same level of evidence generation that we've done for many of these other biomarkers that have come before. You think about amyloid positivity in CSF. It's taken us 10 plus years to get to the point we are today. And I think, similar efforts to advance, to standardize, to ensure that we understand the biology. It's not just about the analytical validation pieces, but also the biological and the clinical validation of these. And I think that's where, as Martin was saying, collecting these samples and these biomarkers, in the study, even if they're not utilized for patient selection at the beginning, we're building a body of evidence now. And so we need to be very thoughtful about how we include those and do these post hoc analyses to better inform on what the utility of these biomarkers will be in the future.
Thank you, Carrie. And I'm mindful as you make those comments. I think sometimes if we're talking about collecting samples at scale in more real world populations, we do have to be having these discussions with patients and the public about why we need to collect these samples and why we need to start making these shifts in our clinical pathways. So Sam, I'm sure you're well placed to comment on that from AR UK's point of view.
Yes, certainly. So as the non-medic among us, my focus has been on understanding what people affected by dementia and the public more broadly think about these sorts of issues. So, for example, in a piece of work we did a couple of years ago, we found that only 40% of people would be willing to undergo a lumbar puncture in order to understand their risk of developing dementia. And so when you compare that to, say, taking a blood sample or having a scan, which is more like 80%, we can see that there are real challenges in participation in clinical trials more broadly, where people are sometimes put off by the invasiveness of, for example, accessing CSF. But obviously, we can't hold back on things just because it's difficult. And we know that, for example, in the rest of Europe, there are different attitudes towards lumbar punctures, for example, and it's very much driven by clinician confidence. So I suppose the challenge for us is really about how do we increase participation in clinical trials. And so part of that, as I think John described in the last session, is about better marketing, ensuring we have a wider pool of people who can be involved in clinical research. And I think actually taking obviously risk data, perhaps genetic, and then blood samples is actually very easy and not costly. So that seems like a better starting point to ensure that you have a general sample, and then think about, for example, how we can filter more effectively and hopefully reduce our screen failure rate by funneling people in a different way, but overall attracting more people to participate in clinical trials.
Yeah, I'd completely agree with that. And I think it's interesting that a lot of the discussions in clinical services about the type of pathways that we need and the data we need to collect, including biomarkers or blood samples, is being driven now by the advent of disease-modifying drugs, or the recognition that those are very close. However, I've also spoken to clinicians and researchers very recently who have commented, well, we're not going to be able to change the services until we have the drugs, which in my mind means we're missing a huge opportunity to collect data in the interim, because most people in this population clinically and their families, in my experience, are also incredibly keen to get involved in trials and will do anything they feel would be useful for the future knowledge base. But I do think it's about having that conversation, and I don't think we're having a big enough conversation yet in the public, if I'm honest. And again, kind of on that, to add on to that question, I guess, one of the things that I know clinical researchers get concerned about, and certainly clinicians, are the ethics about collecting samples from people, when A, we may not be clear what we're going to use those samples for, and B, not being able to necessarily therefore feed data through about what that means in terms of somebody's risk, prognosis, diagnosis. But also, while we do not have disease-modifying treatments currently licensed. And again, my sense is amongst clinicians and clinical researchers that people are becoming less paternalistic, if you like, about this, and more aware. Again, we need to have a discussion about what the public and patients want to do and what information they want. But any comments from the panel about the ethics of collecting, of biobanking samples at scale?
I suppose I'm happy to jump in on this one. I was just going to say, we've been doing some work on this recently to understand people's preferences around diagnosis and diagnostics, but also the principles of earlier detection. So many people understand that in cancer, for example, early detection leads to better outcomes. But obviously that's because we have disease-modifying treatments. In Alzheimer's disease and dementia, that doesn't follow. So the principle of early detection still stands, whether it really resonates with the public, but it feels potentially more difficult to the clinical community because then there isn't that offer of anything further than symptomatic treatments and post-diagnostic support. Whilst people do value the ability to plan and make different decisions about their lives, I think the offer is less compelling, and we still know that many people don't come forward for a diagnosis because there's a fatalism around dementia more broadly, and real stigma and fear around the condition. So there's a lot that we need to do to challenge that, but also convince people that participating in clinical research is also part of the answer. I don't think we have the same culture that they do in oncology, that where people have metastatic cancer, their clinicians are often offering them opportunities to participate in clinical research. We don't see the same in dementia. And I think, Vanessa, you were commenting earlier that you're finding that the age of diagnosis is actually getting later and later, whereas with the advent of disease-modifying treatments, we want to be identifying people younger and younger. So I think there's a real challenge there in terms of reaching the right people and then funneling them, ensuring that there are the right routes to ensure that people can participate in clinical research.
Yeah, just to come back on that, I do agree with that. And I think I've certainly been in meetings recently with patient and public representatives talking about things like brain health clinic initiatives, which are very much looking at how do we start to change the clinical pathway infrastructure to gather these types of markers at an earlier stage so we understand trajectory of disease, but we also recruit more efficiently to trials. And a lot of people were commenting those meetings that, well, at the moment, we can't support clinically the people who already have a diagnosis of dementia. So why are we spending resource on identifying people earlier in the disease path? Well, when there are no disease modifying drugs. So I think we have to be really mindful that, from my point of view, to be really frank, I think this is an area that has been underfunded and underserved for a very, very long time. And we've let our clinical services dwindle to a bare minimum where people have very minimal biomarkers, including imaging done actually across the board. And so we need to pull back from that and explain to the public and patients why we need to be changing our pathways at the moment. Tor, I think you were going to comment.
I mean, it's a question that I've had not uncommonly from ethics committees. What are you going to do with the stored blood sample or the stored CSF sample? We understand that you're collecting it for exploratory purposes. You might use it in the future, but we'd like a little bit more detail around that. So I think, you know, it's more than just engaging with the lay public, it's also engaging with professionals who are overseeing the conduct of clinical trials as well. Yeah, I mean, the narrative around the purpose of biomarkers is particularly, you know, when you're diagnosing people maybe 10 or 15 years before clinical dementia, if that's the intent, is going to be critical, as Sam pointed out.
Absolutely. And just moving on from that, one of the other things I know that we wanted to raise as a panel was also thinking about genetic testing as well, and how genetic risk is incorporated into any markers that we are assessing, again, both in terms of understanding clinical prognosis, but also recruitment into trials. Again, obviously, if we're talking about saliva testing, for instance, for genetics, that's actually an incredibly easy thing to already do in clinical services. And again, a lot of the brain health clinic services that are doing this already, that again, as I said, are trying to address how do we gather more biomarker data in earlier populations. I think one of the things, again, I've heard a lot from clinicians is they are very concerned again about how do we feed back information about genetic risks to patients. This is not something that obviously they're used to having a conversation about. Also, obviously, because most people come with carers, with family members that are also concerned about how do I understand that risk in the context of the wider family. So any thoughts from the panel about the utility of the genetic assessments, but also how we manage that in terms at the scale on with a larger population?
Maybe I can offer a couple of thoughts here, and I'm going to start off just by coming at this from a drug discovery perspective. I mean, we're, you know, GWAS has been the kind of standard method for identifying disease risk genes, and, you know, beyond mutations in amyloid precursor protein and the presenilins and the ApoE epsilon 4 allele, there have been at least 20, maybe 25 different genetic risk loci identified in recent years as risk factors for AD. So the question is, what do we want as drug developers this information for? And I think the hope is that we can translate the genetic findings into an understanding of the functional mechanisms underpinning disease pathogenesis in AD. And, you know, this may throw up some validated drug targets that could potentially turn into effective therapeutics in the future. Those are the high-level objectives. And from my perspective, I would again frame it around the narrative. You know, what is the objective of collecting this genetic data? But I'll maybe leave it to someone else to build on that from the you know, from the you know, how to relay that to the lay public.
Sam, given that, I don't know if you want to come in on that point.
Who did you say? I'm sorry.
Sorry, I said Sam there, but I obviously didn't say it clear enough. Apologies. I was just thinking again about how we have that discussion with the lay public.
Sure. So in a previous role, I worked at Breast Cancer Now, and there was some work to be done on BRCA testing and a real sense of a lag time between someone in someone's family realizing they carried the mutation and another person in the family being ready to be tested. And I think there was much more awareness in that community around the genetic risk factors, whereas I don't think in Alzheimer's disease there's really much of a sense of the range of genes that are involved and the likelihood of developing Alzheimer's disease if there's a kind of genetic or family risk. You know, similarly, I think there's an underdeveloped understanding of modifiable risk factors. I think risk generally is quite poorly understood. So it does feel as though there's a huge job to do in educating the public to understand some of the challenges, but also to engage with them. So obviously modifiable risk factors are within our control to some extent. So it feels as though all of the emerging insights we are developing help us to have different conversations with people. So how can we start having those conversations in midlife so that people can start taking preventative actions? And how can we use some of these insights around either behavioral risk scores in terms of lifestyle and other factors, or including genetics? So programs like Our Future Health, for example, we may be able to get a greater insight into some of these areas. And it feels like polygenic risk scores might be a way of having those earlier discussions about stratifying risk and signposting people into different actions or interventions that might reduce their risk.
Oh, absolutely. Any other comments from the panel? Carrie?
Yeah, I was just going to add on this point. I think, you know, Thor touched on this a bit in terms of thinking about the utility and the value of biomarkers and how important it is to have the support of the in the context of clinical development, the you know, the site investigators. I think this is similar. You know, we really need the education to come from there as well. But it's not just about the principal investigators. I think in the context of the genetic screening, we also really do need support in terms of genetic counseling to help, you know, not only the patients but also you know, their primary caregivers and family members, right? I mean, once they receive this information, knowing carrier status, it brings a bit of responsibility to them where they feel, you know, necessity to inform family members, helping them have the information in a way that's understandable, but also in a way that they can communicate effectively with those family members is really quite critical here.
No, I would agree. And I mean, it makes you wonder, as a sense, with some of the brain health clinic initiatives, which are at the moment variable in terms of the population they're aiming their services at. So certainly the one we have in Oxford is looking at people already referred to memory clinics. But I know that different approaches are being taken elsewhere in terms of trying to identify populations earlier in the disease trajectory. But whether, as you said, should we be focusing particularly on people in that preclinical group, that kind of midlife group, and emphasizing the importance of getting some clear assessment of risk and how you manage that in a very kind of proactive, positive way, that actually there are things you can do at this point, even ahead of any disease modifying treatments being licensed in the UK. Talking about that, I mean, what does the panel think about the process of how we validate biomarkers? And I guess along from that, drug targets as well. I mean, Thor, you touched on this a little bit earlier in terms of the process that we follow earlier in trial design to do that. But any thoughts on whether we should be developing a new pathway for validating these biomarkers more efficiently, so that we are not waiting for long periods of time to be sure as to what biomarker we need to do in which member of the population?
Well, I'm going to just refer back to Charlotte Toynason's great talk this morning, where her lab is really focusing on the development of novel biomarkers using proteomics approach. And it seems to me that understanding the CSF proteome changes in AD and other dementias is quite important, so that you can distinguish between the different dementia subtypes, MCI versus AD and AD versus controls, and also come up with the appropriate cluster of biomarkers that will reflect different biological processes on the trajectory of the disease, accommodating all possible mechanisms, oxidative stress on one hand, mechanisms related to protein clearance, lipid metabolism, synaptic dysfunction, cellular remodeling, immunity, maybe vascular function as well. And lipids also came up in the discussion as well. So, certainly biomarkers that address all these possible mechanisms would be important and may give us insight into new targets. And incidentally, drug developers have traditionally talked about one target, and that's been the conventional approach in drug development. But I think we have to acknowledge that's probably going to be quite absurd in a complex disease like dementia. We're talking about multiple mechanisms. It's going to probably need multiple drugs targeting it, as we know from other disease areas: cancer, heart failure, asthma, hypertension, in the late stages.
Indeed, and I'm kind of struck by some of the things Martin mentioned this morning in his talk about the need for lengthy follow-up sometimes to be able to see that target engagement and change in symptomatology and progression of disease. And I think I made a comment at the time that I'm aware one of the real issues we have with that is just having clinical follow-up for patients who at the moment come to memory clinic services at all. And so similarly, we're going to run into the same issues for people in the preclinical group if we start assessing people earlier. So maybe we do need to start thinking about multimodal biomarkers, but also how we can gather those novelly. And I don't know whether, again, somebody actually has raised in the chat, and I think it's good to interject this at this point, what the panel may think of adding in things like digital biomarkers, if we're thinking about a multimodal set of biomarkers that's useful to identify or track disease and response. Any comments from the panel? Carrie, I think were you going to say something?
I was just going to say that I do think the integration of digital biomarkers is going to become incredibly important. I mean, I think they offer us an opportunity to collect data outside of the clinic often. And that's really critical in terms of, increasing accessibility and ease of use, as well. And I think that will really provide great utility in a much larger sample numbers, right, than we can really gather in the traditional biomarker collection that we've been able to do thus far.
Yeah, completely agree. It feels as though both for early detection, but also monitoring and management, for too long it's felt like there's nothing that people can do themselves. And, many of the people we speak to are madly doing Sudoku and, furiously crosswords and, whatever they can to protect their brain health and to keep their brains active. But actually, a validated tool that's used within the NHS would give that credibility, but also support clinicians in making diagnoses as appropriate, but also monitoring people. I think there's a real sense of, particularly around MCI, some people never get a diagnosis, some people get a diagnosis, but it's, but there's no follow-up. And so I think being able to potentially monitor progression or be able to describe that there is no progression and therefore that's a steady state, I think is really helpful to people, gives them some sense of certainty or being looked after in a way that they don't currently feel.
Yeah, I don't know, Martin, if you wanted to comment, because again, I think I raised this question with you about the fact that gathering data long term can be a challenge for people with dementia or at risk of dementia, just because they're often actually not in secondary care, and even in primary care, they're not seeing their GPs very often, as you said, other than when they go to hospital for hospital admissions. Obviously, that's a great opportunity to understand are things changing, but then you're dealing with people that are old with complex medical pathologies and comorbidities where it can be difficult to tease out how that relates to their dementia pathway. But any comments on how we go about gathering these data long term?
Yeah, sorry, I just missed a bit. My laptop decided that it would turn itself off without me touching it. So apologies for that. So the key thing is not losing people. I think I said that earlier on today. In long-term studies, people tend to move around. They move around physically, whether they go, they move to be nearer their son or daughter, whether they move to go into a nursing home. They also move their clinical care around, whether between different GPs, between different hospitals, between different parts of the healthcare system. So the number one challenge is actually keeping track of the individuals, so that if you do want to know something, you can actually get hold of them. Actually, the NHS is pretty good at that. It's not perfect, but it's pretty good at that. The second thing is thinking about what layers of information do you want, and for example, even for the sort of studies we're talking about now, thinking about the big warning signs when it comes to safety, death, major reasons for admission, major complications, and so on. The third piece is thinking about medication use or other investigations people might be having in primary care that might actually be indicators of something's changing. And then one gets into the more, if you like, more subtle things, which it tends to be around some combination of symptoms and function. And at that level, the question is, can one use some version of patient-reported outcomes over the longer term? When I say some version, what I mean is they're sort of actively patient-recorded outcomes. Open up an app and press a button and say, with a happy smiling face on or something else, and passively collected, looking at the amount of activity people are doing, how much walking they did yesterday, and so on and so forth. Again, some of the difficulties with those is perseverance with the device. I mean, I don't know how many apps you've got on your phone that you downloaded with great enthusiasm and used for about 3 weeks and haven't used ever since. There was a study in the US called My Heart Counts, which was launched at an Apple sort of annual launch day. And I think it had 40,000 people logged on in the first 2 hours and registered for this trial. And if you looked on day 2, it was down to about 400 as far as I could make out. There are real challenges with that, some of those sort of technologies and perseverance. But again, it keeps coming back to, do you know where these people are? And getting some information that gives you a sort of a signal that there's a problem, that there's a problem, then allows you to dig into that problem in more detail. But I think that's probably as much sort of general thought as I can give without thinking about any specific trial or specific situation. But probably the good thing in this particular context we're talking about now is a lot of these are not quite target discovery trials, but they're early phase trials that we're talking about, where actually one's talking about relatively short periods of follow-up, at least in the first instance, to try and identify the short-term effects about whether a drug has any potential or not. I can see Tor's got his hand up, so I'll pass over to him.
It just reminded me of, I mean, the movement disorder specialists are very interested in this area because they see obviously actigraphy as one digital tool that can be used to monitor their patients in the real world. And it reminded me of what Ken Marek at the Michael J. Fox Foundation described as the free-range human experiment. And there's quite a lot of advanced thinking in the movement disorders areas, but I do fully appreciate the challenges when you're moving into more cognitive problems that dementia patients have.
Thank you. And one thing that strikes me also is about how we may use those slightly more novel markers, if you like, to improve the diversity and representativeness of the samples that we recruit to trials. And again, I guess it's that thing that's come up a few times today about the need for homogeneous populations in trials versus the fact that people with dementia are very heterogeneous, and actually the people that are often very vulnerable are the people that don't engage well with services and certainly wouldn't engage well with apps or may not be able to access that technology. Again, I don't know if panel have any thoughts about whether using more novel biomarkers, digital biomarkers, would enable us to increase our diversity, or whether it would just reinforce the issues we have about representativeness. Sam?
So we know that socioeconomic deprivation is often an indicator of later, less accurate diagnosis. So in a brave new world of blood-based biomarkers, if a GP, for example, were able to take a blood test and provide someone with an indication of dementia risk, might it overcome some of the actual social barriers that currently stand in the way of someone receiving a diagnosis at the right time? I think it does herald a different way of patients accessing NHS services if those discussions can be brokered either as a routine health check in middle life or later on as a result of a simple blood test. I know that's moving away from the clinical trial space into how we might implement breakthroughs into the NHS, but I think it does herald an exciting opportunity. Obviously, there's a lot of concern at the moment around AI, so thinking about digital biomarkers and whether we replicate some of the biases we find in society. But I think if we're mindful of those biases and we seek to enrich our trial population so that we have that diversity, and I think that's mostly done through outreach, specific programs to reach those communities that are seldom heard. I was very interested in Martin's presentation earlier, the way in which by recruiting all over the country, the diversity of the trial reflected the diversity of the UK. And I thought that was a really powerful case study of the ways in which widening participation really means that your trials become much more representative.
Yeah, indeed. Carrie?
Yeah, I was just going to take it back to the point Sam made about blood-based biomarkers and just say that I do think there's obviously great opportunity to improve diversity recruitment to clinical trials there with these types of screening methodologies. I do think there may be some limitations even there in terms of the sites and PIs, operational things that we'll need to overcome to really see those realized. But right now, I think there's still sort of a paucity of data around the blood-based biomarkers in these underrepresented populations. And so to really be able to think about utilizing them to inform on populations to select in particular in these groups, I think we're a little bit ahead of ourselves there. And then two, I think in terms of the real world, yes, I think there's opportunity to impact, probably speed up enrollment in trials, particularly in these populations. But I think the real value will come to when we have these assays, these methods for these different blood-based biomarkers adequately validated so that they can support these real world applications. I think that's really where we're going to see the great benefit.
I think you're right, although interesting, Masood Hussain also put in the chat that digital cognitive measures are going to be fundamental for patients. Obviously, patients don't always care if their plasma or CSF biomarkers are going up or down, but they do care about their cognitive performance, and that can mean real things for real patients and families. And I think that's very true. Again, it is about, I think, again, public, patient, wider engagement and education around why we need to be doing these plasma biomarkers as well as others. And again, I've been in a few meetings recently where we've been talking about the potential for disease-modifying drugs and what information would patients and families want in terms of the likely efficacy of these drugs, in terms of, are we talking about an improvement in cognition? Are we talking about a stabilization? What would make a difference if you were offering this drug and you wanted for a patient to consider taking it? And I think, again, we don't have these conversations often enough. I think lots of people have different views as to what treatments or management tools they would want, and what biomarkers they would want to undergo. But if you explain clearly the benefit of doing these things, most people are going to want to be engaged. But I like Sam's kind of comment about maybe we need to be thinking about earlier screening of sorts, if you like, to encourage people from the outset about getting their real world data collected and engaging with the idea that brain health is something they can be empowered to maintain and to monitor over their lifespan. Another question that came up from the group was how we could use biomarkers to understand the specific vulnerabilities of areas of the brain to things like Alzheimer's disease. And the fact that, again, we know this is some of the advantage of using things like PET, for instance, to understand how the pathology affects different areas at different times in the trajectory of the disease. Maybe things like plasma biomarkers aren't going to be as sensitive for that. Again, any thoughts about the biomarkers we're considering now and the utility of those in trials if we want to understand the mechanism of the disease and its pathway? Carrie, I don't know if that's a legacy hand or a new one. Legacy hand. Any comments at all on that question?
Maybe I can offer some thoughts here. I mean, I think if the specific question is around, you know, the long-standing conundrum of, you know, why is it that some AD patients have normal cognitive function, but also have AD because they've got a large amount of A beta in their brain deposited, and why some people with phenotypic Alzheimer's disease have little or no plaque deposition or tangle deposition. If that's what the question is, why are some patients more vulnerable? Why in some brain regions more vulnerable than others? Why is it that the cortex and the amygdala and the hippocampus are vulnerable, but you don't see changes in, say, the cerebellum or the spinal cord or in the brainstem? I mean, if it's a question around what are the differences between the resilient and the vulnerable neurons or brain regions, then it seems to me that the way to approach this is through perhaps understanding the differences in the molecular signatures expressed on the different neuronal populations in healthy brains versus AD brains and vulnerable versus resilient regions. And this will arguably give you a better insight into the spatial and chronological events in dementia. But it will, of course, require that we think carefully about how to integrate data from all the different sources and research groups globally, and overcoming barriers to data sharing and data accessibility as well. But it's about interconnection and interoperability and linkage of different data sets. And I think NIH, they had a AD summit 3 or 4 years ago, and they were talking about developing an AD connectivity map based on omics expression signatures and different disease relevant cell types, and that could help to systematically map resilient versus vulnerable brain regions and track the trajectory of the disease. And ultimately give us better insight into mechanisms of disease. Added to this, there's a need to understand, as Charlotte was alluding to this morning, a better understanding of what's going on in glia, not just neurons, and also vascular changes as well. But that's maybe a start there.
And thinking about that, I know we've talked quite a lot about blood biomarkers, but obviously there are things we still need CSF for, as you said, particularly maybe thinking about microglial responses, inflammatory responses, etc. And again, I'm kind of struck as a clinician that, as we said earlier, the vast majority of patients coming through memory clinic services, or again, if they're being referred earlier, people who maybe in the future will not have access to CSF testing or PET. And I wonder, again, I'm thinking about the Brain Health Clinic group that Oxford is part of. We've had quite a lot of discussions about whether we should be focusing on developing infrastructure to deliver blood-based biomarkers, or should we actually be biting that bullet and saying to patients, in certain cases, we do need to think about CSF testing. As I said, that's very much from a clinical diagnostic point of view, but also encouraging understanding amongst patients from a clinical trials point of view why those biomarkers may be useful. I don't know if people have thoughts about that. Again, I'm saying this just in recognition that a few years ago when I was involved in the EPAD study, I went to a clinical research site and talked about the need for CSF testing as part of that, collecting that cohort. And there were staff, and this was clinical staff, gasping in the room at the prospect that I would suggest people needed a lumbar puncture to collect data. So this is not an easy question in terms of how we work with most clinical services in the UK. Sam, you're nodding your head.
Yes, vigorously. It's something we've been thinking about a lot. We published a report in 2021 looking at the short term for diagnosis and diagnostics, and also the medium and longer term, and whether you would ever get to a position where blood-based biomarkers might leapfrog CSF and PET. Because obviously, if we did get a DMT in the next couple of years, approved in the UK, you would have to administer it based on what's on the label. So you would have to probably give people at least a PET scan or a lumbar puncture, which is a complete transformation of the current care pathway and would be hugely difficult to implement. So I suppose the question to government is whether, if they invested in blood-based biomarkers now and validating those, whether you might leapfrog. So essentially go through the Betamax stage and straight to VHS, but whether that's even desirable. But I suppose we also did a piece of work with the Royal College of Psychiatrists, and only 10% of people have access to those molecular diagnostics. So clearly there's a huge job to do in terms of both access to literally enough machines so that people can both have access to conduct PET scans, but also access the confidence to interpret the results that they receive. So yeah, a huge job of heavy lifting, but I feel that clinical researchers can be preparing the ground and sharing those insights so that the wider field and NHS can be preparing both clinicians and patients for what the future might hold.
Yeah, I know John Gallagher certainly raised the issue, didn't he, about neurochemistry as one of those issues we have across the UK in terms of access. But I would agree. And I mean, even when you're thinking about simpler modes of imaging, if you like, I'm aware that in certain areas of the UK, and one sits within the Oxford region, commissioners, the services they paid for memory clinics, at the moment they do routine imaging only on 10% of the people coming through their clinic. So that's even CT, let alone MR. So we have very little baseline data often in this population, but I think you're right, Sam, it's about emphasizing the importance of this and how that may help us validate biomarkers that are then going to be much easier to deliver when we do absolutely need them and we need to be allocating disease-modifying treatments. Any other comments on that?
I mean, I just comment, I think it really is about thinking towards the future. Yes, the blood-based biomarkers are the direction that we would like to be going, for all the obvious reasons in terms of cost, accessibility, but we can't put aside these other biomarkers that we put so much effort into already, and are really quite critical to getting those blood-based biomarkers to the stage at which we need them to be. We need to understand how the blood-based biomarkers relate to the imaging, relate to the CSF, and so the focus can't be so pointed onto the blood-based biomarkers that we forget about those really important pieces to the clinical validation of the blood-based biomarkers themselves.
Yeah, I think again the comments Martin has made about just trying to think about the routines of biobanking samples across clinical populations is really powerful, and I think it is the kind of thing that both patients and clinicians would engage with, but it's not something that we've even thought about doing. As I said, we've not had the conversations with either group to date, and I think most people would be on board with that. And if you start having conversations with patients and the public about the need to do that, say with blood and plasma, then it's easier to then have a follow-up discussion and say, actually, this is the rationale as to why we may need to do CSF testing in some people. Again, it's just, I think about expectations. About a year ago, I remember somebody's relative, for someone who was referred to our memory clinic, called us up and said she didn't understand why her mother had to attend the clinic because couldn't we diagnose dementia via questionnaire? So, people's expectations of what dementia is, and I still think that's an educational issue about people still to a certain extent thinking dementia is part of an inevitable aging process and not something that is a separate pathology in itself. I guess I'd like to, I don't know if there are any other, if people do want to put questions in the chat, do. At this point, we're coming up to the last few minutes of the session. So if people have any other questions for the panel, please put those in the chat. I guess in terms of something I'm very struck with when I go to these types of meetings where we're talking about things that maybe need to shift in clinical trials, we're often left thinking, well, what are we going to do first? What are we going to do next? Because there's so much to shift, I think, and so much potential. So I'm going to give the really hard question to the panelists and say, what do you think the action plan should be at this point? What should we be doing? I know we can't all do this individually. We need to do this collaboratively. But what would be your priority, I guess, in terms of biomarker work at this stage? So I don't know who wants to go first with that.
So mine links to the Conservative Party manifesto of 2019, where the government promised to speed up progress in clinical trials. And so how we've interpreted that is really thinking about what would transform the clinical trials landscape in the UK. What is the infrastructure that is required to ensure that there are more people participating in dementia research, that we're reducing that screen failure rate. We're ensuring that we have cohorts that are ready to go. We're attracting industry to look at new molecules to ensure that people in the UK are enrolled to those trials, and that we're ensuring that patients in the NHS can access new treatments of tomorrow. And I don't think that's too far-fetched. We've seen through the COVID vaccines task force how a clear mandate with a direct line to the Prime Minister and a real focus on delivering outcomes can really deliver that transformative change in a short period of time. I know that was an emergency situation, but if you ask me, the challenge we're facing with dementia is a similar challenge, and it requires an urgent response.
Thank you, Sam.
I know that's not just one thing.
Sadly, I think not one thing is going to do it. So absolutely. Other thoughts on plans of action from here, if you were Martin?
Yeah, I'll build on Sam's comments, really. I think that the ways of doing trials has to be easier to do the trials which will provide you the information that will actually change both decisions about drug development and about drug usage or not. And that's not the framework we've got at the moment. That does require certain parts of, if you like, infrastructure. It requires partnerships, that hence my comments earlier about industry, academia, charities, and the health service, conversations with patients and their carers and relatives. But it also requires really rethinking about what the rules are that go around trials, not to make trials worse, actually to make trials better. And just because you can check something doesn't mean that you add quality by doing so. And I think we've got to actually be looking at how can we frame regulations and guidelines that would actually help so much of this. And no one organisation can do that. That has to be done together. But I think getting the rules right is going to be really important.
Thank you. Yes. Tor and Carrie, what would your manifesto be?
Well, I mean, how can I build on those two wonderful sets of vision? I mean, I completely agree with, the public-private partnerships and industry and the health service talking together. And I would love to see, better clinical trials. To date, from an industry perspective, they've largely been very kind of process-driven and traditional phase 1, phase 2, big phase 3 does anyone want to invest in phase 3? Because then you're really talking about, you know, a 10 or even 100-fold increase in investment, big risk. And, you know, some of the costs are beyond the reach of even some of the biggest companies like Lilly, Biogen, Roche. There are not many big players in the field. So, you know, the whole decision-making process of how to make that leap from the smaller clinical pharmacology stage studies that might look at biomarkers and might do studies in well-defined cohorts. How do you make that leap into phase 3? I don't know what the answer is. I'm not sure I've heard the answer today, but I think certainly with the help of the partnerships, everyone can work together and figure out a more realistic and credible decision-making framework for progressing new therapeutics. That's my vision. I'm sorry if it doesn't offer any clear answers at this stage. It's a difficult question.
It is a difficult question, and I think that's absolutely the case. We just need to work through it together. Carrie, did you have any final comments probably at this point?
Agree completely in terms of the comments that others have made, particularly the point around the necessity really here for these types of partnerships. I think thinking, specifically about one area where that's critical, it's kind of asking questions like this. I think we really need to work carefully to align on purpose. Very narrowly, if we think just about that question around blood-based biomarkers, I think one of the challenges that we have right now is there's so much excitement. And I think, many potential purposes for these. Even if you think just about patient selection, are we thinking about utilizing blood-based biomarkers for amyloid positivity, tau positivity? Is it both? And I think the sooner we align on these types of questions, and these are probably true outside of blood-based biomarkers as well, the more quickly we're going to really be able to advance the field. And clearly this is, one of the specific purposes of meetings like today.
Thank you, Carrie. So on that unanswerable question, at least to date, I'm going to hand back to Masood. I think we have another plenary session at this point. So thank you all to the panelists.